gentlemen, the books is in. The books are in. Yep. They in. They're here.
Log in to KofiPaisaTV.com and get Spears of the Mossy Volume 1 by Kofi Paisa Research Team, from Spears the Pen Volume 2 by Kofi Paisa Research Team, Spiritual Combat Volume 3 by Kofi Paisa Research Team, One Night Before Bed by Any Harek Shun Kalfunny, Kalawa's Trip to the Motherland by Kofi Paisa, From Cocaine to Consciousness by Any Harek Shun Kalfunny, As I Learned, We All Learn by Kofi Paisa. Also, if you want fresh merch, go to our online store, Family Connect Apparel, and get all the brands. Also, get our 54 deck of quiz and educational cards available now on KofiPaisaTV.com.
Copyright disclaimer: under Section 107 of the Copyright Act of 1976, allowances made for fair use for purposes such as criticism, comment, news reporting, teaching, scholarship, and research. Fair use is a use permitted by copyright statute that might otherwise be infringing. ETM Hotel, Rennie, Sean, welcome and peace. My name is Sean. I'd like to thank everybody for tuning into Science with Sean. I sound a little bit better on the microphone because I had to change the direction. You're tuned into this episode of Science with Sean. This is our session 3. Hopefully we're going to knock this out and make this the last session from a study session that we had the other day. We just want to follow up, get through the exercises, and we got class tomorrow. So we're pretty much just doing a live demonstration of this to keep everybody abreast of what we're learning in our biology class. And hopefully if you're interested in anything like that too, you'll join us. But, you know, like I said, this is a journey. Some of us have taken upon ourselves and to try to be better students, be better researchers, be more scientific, and help advance scholarship. So in this, we're going to take our time out to learn more about transmissions and transitions and transmitting. I had to throw that in there like that for a particular reason because, let's cut straight to the chase, right? So, let me fix my mic again. There we go. I need to get right here. All right. So I do want to say peace to everybody in the chat. Please like the show, share the show, tell a friend that we're live. And I do appreciate all the views that we have gotten, and the people that have participated in the class. In the description of the book, in the description of the video is the book that we're dealing with, and also the link to what Sars Cov2 taught me. Do I sound a little staticky for any reason? If you could hear me okay, cool. If you can't, let me know. Again, like a little static. I'm gonna try to handle that, make sure that it's not my cord or anything like that. I got my headset in. Probably need to take it out. Okay, you're saying I'm clear. I'm just static in my headphones then. Hold on 1 second, because this feedback killing me. All right, how about now? Oh, okay. Static on the way. Maybe I wasn't up in there all the way. All right. So, come on, mouse. Here we go. We'll go ahead and share my screen. We'll get right into this study group. Man, as you come in, please like the show, share the show, tell a friend. And we're going to get it in. If you have not purchased your copy of What Sars Cov2 Taught Me, please take a time out. Go get your copy of What Sars Cov2 Taught Me. What are you waiting on? We're in the middle of a pandemic. We got all this misinformation going around. Make sure you got your copy of What Sars Cov2 Taught Me. Oh, shout out, and I'm going to say this too. Shout out to, I haven't looked the brother up or anything like that, but there was a brother from New York last night, purchased 5 copies of What Sars Cov2 Taught Me. He's the second person to do that, by the way. Purchased 5 copies. And I'm pretty sure, well, if you're tuned in, bro, I didn't know if you want me to sign all 5. But I am going to sign 1 and send the other 4 unsigned. I didn't know if you wanted them to be to specific people or whatnot, but if you are tuned in and you are listening, or you may catch the replay of this particular show, you can email us on the site. And if you want me to sign a copy, each copy specifically to a particular person, then I could do that. But if you're okay with them not being signed, I am going to sign 1 anyway. But if you're okay with the others not being signed, then, that's how they're going to come. And you'll be updated with your tracking information as soon as we get everything squared away. All right. All right. So sharing my screen. Just a quick follow-up from what we did the other day. I just want to kind of open up the study group with a question that everybody should know the answer to by now. So what is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? For anyone that doesn't understand what the DS is, I basically just gave it to you. Double-stranded DNA viruses. And as a follow-up, No, I ain't gonna do a follow-up. Yeah, I am. As a follow-up, what does this red marker right here mean? And this yellow box here, and this green box here, and this blue and light blue box there. So we have the initial question, right? We have this to start the study off. What is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? And then we have a follow-up, which is what are the two light-colored, and as far as representation, what is the darker and lighter blue represent? The green and the yellow and this red, what does that represent? And when you give me the blue, I need to know, give me an example. I mean, give me more than just this represent. Well, you got to give that explanation anyway, but this represents what and this represents what? This isn't in the book. This isn't in my book. This is in the virology, Principles of Virology book. So I know there's a delay. So I'm gonna wait and see and let everybody answer. I see one answer in the chat. I'm just waiting to see if there's any others. All right. So what is the first biosynthetic event that occurs in cells infected with double-stranded DNA? That's the first question. The follow-up is, in this image, what does this red arrow represent? What does this yellow box represent? What does this green box right here represent? And then the blue and light blue, what do they represent? I'm gonna wait. I only look like one person in the chat trying to explain it. Hey, don't let your head hurt. We all here to learn. This is a study group. Everybody don't know everything. All right, so it looked like we're gonna ride with Nunu on this one. But Benita answered it correctly. She gave a transcription for the question. The answer was actually the answer was on the screen that we was just on. But what is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? Is transcription, B. All right, and in the image that I had here, as you can see, a regulatory sequence in transcriptional control region. So just to let you know what we're talking about as far as this build. Now, the red arrow, correct? No, it does. Hold on. Okay, she went to her notes. All right, so the red arrow, yes, represents promoter. This is a promoter here. All right. The yellow represents indicator. This is an indication. The green represents the Tata sequence. That's correct. And this right here is DNA representation of the adenovirus. This is the DNA plus strand is the darker blue, and the DNA minus strand, the DNA plus and the DNA minus sequences. There you go. And then these colors right here, they represent certain things too. Now, these are basically what you're looking at is protein sequences. All right. So that just kind of bring everybody up that hasn't paid any attention or tuned into the show. This is just something that we're learning in the class that we're actually taking, and virology class. And what we're going to do is we're going to continue, and we're going to get further into it, and we're going to skip through some of the hesitations in the video, and we're going to complete this study class today. All right. So let's go ahead and continue where we are.
It's not really a biosynthetic event. Membrane fusion doesn't involve synthesis of anything new. DNA replication.
Now I want to go back because we asked Dunk this on another show, and he's gonna go through the other answers that was on here that wasn't the right answer, but he's gonna tell you what they are. So make sure that you're paying attention to that, and let's have some fun. Science with Sean.
Okay, so let's get back to the quiz. What do we have? Almost everyone answered now. The answer is transcription, the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses. So the genome is ready. It just has to be made into mRNA because you need at least one, sometimes many proteins. So some people said membrane fusion. It's not really a biosynthetic event. Membrane fusion doesn't involve synthesis of anything new. DNA replication cannot occur until a protein is made. So by definition, you need to have mRNA synthesis. Protein synthesis, now you need to make mRNA before you make protein, right? And that's a biosynthetic event, and it's not all of the above. Hope you understand why. Now, this is a nice summary of the different strategies for transcription of viral DNA. And we have two columns here. On the right is the virus example, and on the left is the origin of the transcriptional components. Where does the stuff come from that's involved in transcription? And for some viruses, retroviruses with simple genomes, columoviruses, those are plant viruses, RNA viruses with reverse transcriptase, say circoviruses, single-stranded DNA viruses. It all comes from the host. The viral genome makes zero contribution to the transcriptional machinery. And then everybody else here, all the other viruses, sorry, I need to bold this to make it clear because I even confuse myself. Then we have an example of the host supplies most of the proteins, but there's one viral protein that's made that participates in the regulation, right? I'm going to do that right now because every year I say I have to bold this and I never do. So let's do it now. And here we have host plus one, and then this should be bolded, right, to keep it consistent, I suppose. All right. So then host plus one viral protein.
For the people that want to follow along with the book, I'm going to tell you what page that we're on in my book. But I'm going to tell you in the link that Chavis just dropped is a free copy of an older edition of the book. I have the latest edition of the book. So my stuff's more updated. And not to take away from the link that Chavis put in the chat, Chavez. But you want to get this so you can follow along a little bit. So when I refer in the book, you can do it. So if you go to the link to the Google Drive that he just put in there, and you pull up the Principles of Virology book, you need to go to chapter 8 in the Principles of Virology book. Now, when you get to chapter 8, we're about 5 pages into chapter 8. You're going to see hold on. You'll see something that resembles what is, well, in this instance, it's not on the screen, but this is a part of like a different indicator. But I will let you know when we get to where we need to get to, because he's about to go into some things, and then I'll blurt out what he's saying, and I'll catch all of us where we need to be, because it's going to get a little curvy here.
Bacteriophages The viral protein is involved in the transcription of the late genes. And then for all these viruses, the viral protein regulates transcription. We'll see some examples of that. And then finally, there's more than one viral protein involved in these herpes viruses. And then, the last category, I have to bold that one too, right? Yeah. You imagine I'm teaching this for 12 years and I still change? Of course, you have to.
Now, just to bring you up to date, okay, he's reading from table 7.1, but in the book that you have, it may be table 8.1. So that would be the second page of chapter 8. If your book flows just like my book, if you look at synthesis of RNA from DNA templates, if you just turn the page to the second page, after the book introduction and all of that, you will see something that says strategies of transcriptions of viral DNA templates, right? What it says on the screen, and it says origin of transcriptional components, and then it says viruses, and this is what he's summing up right here. Okay, so if you have the book and you have the 5th edition, it is on page 200, chapter 7.
Everything is viral. Was that Lego movie? Everything is awesome, right? Everything is viral. Mrna virus and mpox viruses encode the entire transcriptional machinery: polymerase, accessory proteins, regulatory proteins. Why? Because they're in the cytoplasm. They can't get access to the nuclear bureaucracy. Okay, so that's a cool way to summarize that. All right, let's look at some regulation by now. I've just told you some viral proteins function as regulatory agents in transcription. How does that work? Here are 2 different kinds of regulation that we look at. Cascade regulation first.
All right, now he's dealing with regulation of transcriptional by viral proteins. That's going to be on a different page. Okay. There's too many different diagrams here, but regulation is going to be the example that he have here. Hold on. That's on 211. In the 5th edition, it's on 211. I don't know what it would be on the 8th edition, but it on the older version, but it's literally about 7 pages to the right, 7 to 8 pages to the right, 211, or whatever the page number is. If Chavis, if you're watching and you have the older edition, if you can turn the page to where you see this diagram and you could put that in the chat, people will know if they are using the Google Drive. However, we're on page 211. And if you pay attention to what Nunu just said in the chat, you need to make sure that you understand that some viruses do not go to the nucleus. The viruses that do, if the virus, okay, all right, since you want to be smart, right? If the virus goes to the nucleus, what kind of virus is it? If the virus doesn't go to the nucleus, what kind of virus is it? This dude said it might be around 250. Come on, bro. I'm asking a question right now while he's getting a page for those of you who may be trying to find it if you are using the Google link. But let's go.
What does that mean? It's a cascade, right? Here we have gene X being transcribed. It's a promoter, start site for mRNA synthesis. This is carried out by cellular proteins. We make a viral mRNA. This is a viral genome at the top, right? Viral DNA. We make a viral mRNA. We make protein X. Then protein X actually is needed to activate another promoter on the same genome, the red promoter here, to get gene Y transcribed and to make the mRNAs and eventually the proteins. So without protein X, gene Y isn't transcribed. That's cascade regulation. We're going to see some examples of that today. Very cool. And then we have auto-regulatory loops.
Oh, my bad.
Which may be positive or negative. And here's an example of a positive one. We have a viral DNA with a gene. It's got a promoter in front of it. This DNA gets into the cell. It's transcribed by cellular proteins to make the viral mRNA. A protein is made, which then goes back to the promoter and enhances transcription even more. So you get many, many more mRNA. So initially, the promoter works in the uninfected cell. But once you make a viral protein, you get better and better transcription. Right, so that's positive. Negative, you can imagine that the protein made eventually suppresses its own transcription. Why would that be? Or what would the function of that be? Well, that would be to suppress the synthesis of a protein that's only needed, say, for an infection. So let's go through a couple of viruses and see their transcription.
All right. So before we move on there, I want to go ahead and give a little bit more insight of what viral proteins that govern transcriptional DNA templates. And before he gets to where he needs to get to, I just want to put that right there. So these are patterns of regulation, and that's what he's talking about. So transcription of many viral DNA templates. So this top part, remember, is DNA. These arrows are promoters. There can be more than one promoter on each DNA sequence, these sequences here. All right, this is the positive, this is the negative, right? If you see yellow, you got to think indicator, indicator. What we don't see in this particular cascade regulation is what's missing. Just from the example. All right, this is a question. I'm just throwing it out there. It's nothing serious. But anyway, so transcription of many viral DNA templates by RNA polymerase II machinery results in the synthesis of large quantities of transcripts, in some cases more than 10 to the 5th power copies of individual mRNA species per cell in relatively short periods. Such bursts of transcription are elicited by viral proteins that stimulate transcriptions and establish one of the two kinds of regulatory circuits. The first is transcriptional cascade, in which different viral transcription units are activated in ordered sequence, often accompanied by inhibition of transcription of genes expressed earlier, which is what he has in the diagram. Now, these mechanisms ensures that different classes of viral proteins are made during different periods of the infectious cycle is characteristics of virus with DNA genome. The second is a positive autoregulatory loop in which initial transcription or production of a viral protein leads to an increased rate of transcription of the same viral transcription unit. Shout out to Demar. I see you answer my question in the chat. I asked you about the two different types of viruses out there. You've been reading the book. You know what I'm saying? I could tell when certain people been reading the book. You feel me? Like certain stuff started to stick with them, and that's cool. I like that's dope. All right. So the participation of viral proteins confers a measure of control lacking when the transcriptional program is executed solely by cellular components. The following sections describe some well-studied examples of the regulatory circuits established by viral proteins. Now, the first one that he pulls up here is the S40 virus. All right, so he skips over the HIV, which he may comes back to it, and then he goes to the S40 virus. Let's go.
Suppress the synthesis of a protein that's only needed, say, early in infection. So let's go through a couple of viruses and see.
Now, he is on page 218. If you have the 5th edition, 218. So that would probably mean that he's probably 256 or whatever Chavis put in the chat. It's probably 4 pages to the right of that. But I don't think Chavis put anything in there. He says it might be around, but he never confirmed. What's the answer, Sean? I answered the question. I answered it. If it's in the nucleus, if it's nucleus bound, DNA viruses live in the nucleus. Outside of that, the RNA don't. They don't live in the nucleus. Do RNA viruses live in? Can they be in the nucleus? No. Why? Because if it's a single-stranded, all right. So you have single-stranded and double-stranded viruses. They can be DNA or they can be RNA. But in the instance of dealing with the actual nucleus of the cell, in order to get to the nucleus of a single cell virus can't get to the nucleus unless it is a double-stranded. So it would have to be double-stranded in order to reach the nucleus. And most RNA viruses or whatnot, I don't want to get this wrong, but in most instances, I'll just say this, in most instances, that ain't transpiring like that. So I'm having in the cytoplasm. What you talking about? All right, man, you interrupting class, man. You acting like JJ. No, I'm just playing.
You can imagine that the protein made eventually suppresses its own transcription. Why would that be? Or what would the function of that be? Well, that would be to suppress the synthesis of a protein that's only needed, say, early in infection. So let's go through a couple of viruses and see their transcriptional programs to illustrate this. So here's SV40, the virus with a double-stranded circular DNA genome. You're all infected with polyomaviruses like this one, okay? So this is not obscure, even though this is a monkey virus that is used as an example. Here's a timeline of time after infection. So infection begins at the left. The reproduction cycle of this virus is divided into two phases, an early phase and a late phase. In the early phase, the early promoter is active. This virus has its circular double-stranded DNA at the top here, labeled ORI. That stands for origin of replication. That's where DNA replication begins. We're going to talk about that Monday. There's also promoters here that go in two directions, in the early or late directions. And all that means is that in the early phase, only the early mRNAs are made, and the late mRNAs are made in the late phase. So when this DNA gets in the cell, it goes in the nucleus, the early promoter is transcribed. The early mRNA encodes a protein called LT, large T antigen. I'll just refer it to T antigen, even though they're large, medium, and small for various polyomaviruses. And large T has so many functions. It's going to turn on viral DNA synthesis. And the viral DNA synthesis is actually going to, it also turns on the late promoter. The activation of viral DNA synthesis results in the activation of the late promoter, and then you get late mRNAs made. And LT has many, many functions, as you'll see. So it's a activator of DNA synthesis. It can also activate late promoters. It can also inhibit its own synthesis. So once we have enough LT, we don't need it anymore. It inhibits its own production. And so you may ask, what's the function of having two different phases? Well, the function is early on, there's no DNA. There's no viral DNA except what comes in. So there's no point in making capsid proteins yet. The capsid proteins are actually encoded by the late genes that are encoded and produced from late mRNAs. So there's a period where only large T is made. That kicks viral DNA replication in. Virus DNA replication eventually leads to the turning on of the late genes, and then you can make capsid proteins. That's why it's regulated. Okay? How's the late?
All right, again, LT means late transcriptional phase. Late transcriptional phase, and then you have an early. You have an immediate early, and then you have early, but you have an early transcriptional phase and a late transcriptional phase. Again, it's on the screen. Early, late. If you're trying to follow along with the diagram again, you're gonna have to turn some pages. You're gonna have to go. I basically was on 218. Now I'm on 226 in the latest edition of the book. So 8 pages. You will see where it says initiator binding protein, and it will have this diagram right here, which is what he's about to explain. And I'm gonna give you a little insight before he goes. All right, so this is called coupling to initial rounds of replication. Transcriptions of the herpes viral late genes also requires viral genome replication and viral regulators. And let's see what he says. Let's learn.
Late promoter regulator is shown here. This is a very, very cool mechanism. So this is the late promoter, which is not on early when the virus first infects the cell. Only the early promoter is active, and that's shown in this graph. This is concentration versus time after infection. So you start at 0, and the viral genomes come in. They stay low for a while, and then they start to go up, and eventually, they peak.
I saw it. Yep.
Viral genomes
Exactly. You said you heard some static.
To go up, and from that we produce capsid.
I was hearing static too.
Come together and make new virus particles. So the question, and in this early reach,
let me get rid of these headphones.
Only the early mRNA is made. It's not shown in this graph. So what turns on the late promoter?
Yeah, let me get rid of my headphones. Hold on 1 second, y'all.
Right.
I don't know why it's giving feedback. Oh, okay. I see. Yeah, my laptop fan moving around. So that little feedback that we were hearing, yeah, that's coming from it's coming from my laptop. That's the only thing I can think of. All right, so I shouldn't have no issues now. Let's go.
Here. So here's the viral DNA. It's come in the cell. This is the late promoter shown with the right arrow. And in the early phase of the infection, the E phase, the late promoter is inactive because there's a cell protein that binds the regulatory sequences in this region, and they block transcription. This cell promoter is called IBP, initiator binding protein. It is a cell protein, yellow, binds all these regulatory sites, and that represses transcription from the late promoter. It's a cell protein. I'm saying it 3 times now because people get confused. It's not a viral protein. Now, what happens as DNA begins to replicate? T antigen is made first early. It turns on DNA replication, and you may be thinking, how does it do that? Well, we'll learn on Monday how it does that. And then as you get more and more genomes made, you titrate out the IBP. There's a fixed amount of IBP in the cell. And so the more genomes you make, the more viral genomes, there's now not enough IBP. And eventually you get DNA genomes with no IBP bound. And so the late promoter comes on and it starts to make late mRNAs. How brilliant is that? How brilliant is that, that the late a cell protein, its titration by DNA replication turns on the late promoter. I think that's very cool. Okay. So here's an overview of the entire reproduction cycle. To put this in perspective, the virus comes. Now
remember, initially he showed this image at the 8-minute and 30-second mark. As you can see on the screen in the box below, the same image, he did all of that talking an hour ago in this video, and he comes right back to this and he says functions of early and late phases to delay synthesis of structural proteins until DNA has been replicated. And this takes us through the process of how the early and late phases occur. Now, some of this stuff we should be able to recognize, like LT, late transcriptional period. And we should be able to, we should have like a little general idea. We don't have, we're not trying to be experts, but we're trying to know enough to follow along. So we know what happens in the late. We should have a little idea of what happens in the late, right? And then we should know what transpires in that period. Okay, because he finna progress back. He went forward in the book and then he gonna go backwards in the book. I'm just letting you know.
The cell, the DNA is put into the nucleus. It's ready for transcription. The early mRNA only is made. That mRNA goes out in the cytoplasm. It codes for the T antigens. Those go back in the nucleus and they stimulate DNA replication by a mechanism we'll talk about on Monday. So now you have new DNA made, and eventually titration of IBP allows the late promoter to become active. You make late mRNAs, which encode the capsid proteins. So now these capsid proteins can coalesce around the newly synthesized DNA, and you have new virus particles. And so the key here, what's the function of early and late phases? To delay synthesis of structural proteins until the DNA has been replicated, right? You don't need to have capsid proteins unless you have DNA made. Okay, slightly more complicated, adenovirus transcriptional regulation. We still have early and late phases as we did with SV40. Again, this is a timeline of the infectious cycle. But now we have an immediate early phase, right? It's earlier than early. And then there are three viral proteins here that are governing phase transitions. And so
218.
Immediate early region are genes among which encode the E1A proteins. E1A is made first. That promoter is recognized by the host cell transcription machinery. The others are not. The early and the late promoters don't work yet. But E1A is needed for activation of the E promoter. You'll see the mechanisms of that in a moment, how E1A turns on the E promoter.
All right. So on page 218, chapter 7, 5th edition, he says that cellular enhancers and promoter binding proteins are sufficient to initiate the polymeroviral and adenoviral transcription programs and the synthesis in infected cells of crucial viral transcriptional regulators, large T antigen, which is LT, and E1A proteins, respectively. So when he's talking about the E1A, he is talking about proteins. In contrast, a viral activating protein imported into cells as a virion structural component, VP16, which we saw in the other example, is necessary. Well, actually, we'll see in another one. You'll see necessary for efficient transcription of the first herpes genes to be expressed, called immediate early genes. So you heard me mention earlier, immediate earlier, earlier, and late. So immediate earlier is IE, early is E, late is L. I'm trying to see what else I can give us so that we can make sure that we're on point. That's it.
And then in the E promoter is encoded genes. I'm sorry, in the E gene, which is turned on by E1A, it are encoded proteins needed for DNA replication and the DNA polymerase, among other proteins as well. And then there's also a protein encoded in here, E2, that is required not only for DNA synthesis, but entry into the late phase. And part of that is by titration out of repressors, as we have seen, turns on DNA replication, turns that on and allows for synthesis of the late mRNAs and the late proteins. And the late proteins, of course, encode the capsid proteins. Now you have DNAs made, you can encapsulate them into new virus particles. And there's another protein here, 4A2, which also enhances late gene transcription. So 3 proteins govern the transition. E1A is initially made. It turns on the E region, mRNA synthesis, which turns on DNA replication, and that turns on the late phase, and then a late protein 4A2 further enhances late transcription. Again, multiple phases. Now, let me show you how this E1A protein, E1A proteins, they're multiple, turn on transcription of the E gene, right? Now, in the cell there's a set of transcription factors called E2Fs. They were originally discovered through studies of adenovirus.
Page 224.
Transcription. And so they're called early region 2 factors, but it turns out they're, of course, needed for cell proteins, and we'll talk more about that later. These E2F proteins are complex with a second protein called DP1, which we don't have to worry about right here. So these are transcription factors needed for transcription of the adenovirus early region, okay? However, in most cells which are quiescent, these proteins are bound by a cell protein called RB. RB stands for retinoblastoma. So RB binds this. And so even though E2F can bind a promoter, so let's say the early region promoter of adenovirus, the presence of RB will prevent transcription. And the reason is that the RB protein recruits another set of proteins called histone deacetylases, or HDACs. These enzymes remove acetyl groups from histones. Okay, they remove acetyl groups, and so that wraps the DNA more tightly. So here, this chromatin here on the right, they're the DNA wrapped around our histones, our nucleosomes, which I refer to as cakes. When they're acetylated, when the histone is acetylated, the DNA is open and it's accessible to transcription. But when the acetyl groups are removed by histone deacetylases, these are cell proteins, then the chromatin becomes compacted. We call it closed chromatin, and this reduces transcription. So the presence of RB on the E2F brings in HDACs, which close up this chromatin, so the early region promoter doesn't work. That's where E1A comes in. E1A binds RB and takes it away from E2F. And now there's no more HDAC. The histones can be acetylated and the promoter can be active. This is just amazing. Can you imagine the years of work it took to figure this out? So the viral E1A is made first. No other promoter is active. And then E1A pulls off RB, activates the early region promoter, and then that turns on DNA synthesis. We will come back to this when we talk about viruses and cancer, because this is involved in that. Here's a map of the adenovirus genome. Double-stranded DNA, about 35 to 40,000 bases. Pretty long. It has a protein attached to the 5' end, which is part of the primer for DNA synthesis, as we'll see Monday. But there are multiple transcription units, which means sets of genes that are transcribed together. So here we have the early region transcription unit. That's transcribed first when the viral DNA gets into cells. We have the E1A protein, which we just talked about. There's also E1B. Then we have an early region set of promoters are on the other strand. We have an E2, we have an E3, and an E4. These are all early regions. They're turned on by the E1A protein. And then we have late promoters. And you can see L1, 2, 3, 4, 5. We also have a major late promoter, which makes a huge pre-mRNA that encodes mostly structural proteins, right? Hexon, penton base, fiber. But look at the early region encodes the DNA polymerase and DNA binding protein. These are what gets DNA synthesis going. And then finally, the major late promoter is turned on after DNA synthesis begins. Really remarkably complicated. So here's an overview of this. Here is the adenovirus reproduction cycle with the immediate early, the early, and the late phases. We start with virus entering the cell, which we've talked about. The DNA ends up in the nucleus. The immediate early region is active. The promoter is active. So the E1A and E1B mRNAs are made. The E1A protein is produced, goes back in the nucleus. It binds RB. It allows freeing up of the E2F transcription proteins. And now the transcription of the E2 promoter. The early region promoters can occur, not just E2, but others. And the proteins include the ones needed for DNA synthesis. So now we have onset of DNA synthesis, which is shown here by going from blue to red DNA genomes. And that onset of DNA synthesis allows turning on of the late promoter. Late mRNAs encode the structural proteins, which come back into the nucleus and allow for assembly of new virus particles. So a carefully orchestrated series of events made to optimize materials so that early you only make what is needed. The other point here is that these viral promoters are highly regulated. They're not recognized by the cell. So the E1A needs to kick all that into going forward. And the last virus transcriptional program is for herpes virus. And this is a little more complicated, but there's still three phases: immediate, early, and late, similar to adenovirus. But one twist that I think is really amazing, and that is even the immediate early promoter doesn't work in cells when the viral genome comes in. So we saw for the adenovirus genome, the immediate early promoter works, but here it doesn't. What to do? Well, in the viral capsid, in the particle, there's a herpes virus particle. In this space between the nucleocapsid and the membrane, you can see all this dark stuff. These are all proteins. And among those proteins is a protein called the VP16, which is a protein that allows recognition of the immediate early promoters of herpes viruses by the cell machinery. Without that VP16, virus is dead in the water. So now you turn on immediate early transcription because of VP16. Some immediate early proteins like ICP0 are made that activate early promoters. Those produce mRNAs that encode replication proteins, and then DNA synthesis activates the late transcriptional units, which encode structural proteins. So very much similar to adenovirus, except for this twist of needing a viral particle-associated protein to turn on immediate early transcription. And so here is an overview of the herpes simplex reproduction cycle. This virus fuses at the cell plasma membrane, and the nucleocapsid is transported down to the nucleus. Remember, it docks onto the nuclear pore, and the DNA exits through a portal. It actually then circularizes in the nucleus. We'll talk about this on Monday. But as this nucleocapsid is moving in, all those proteins in between the nucleocapsid and the envelope of the particle, they end up entering the cytosol. And VP16 is transported into the nucleus where it can activate the immediate early promoter. Those mRNAs go out to the cytosol. The proteins are made. They go back in the nucleus. They turn on the early region promoters. Those proteins made start the DNA synthesis procedure, and then DNA synthesis turns on the late promoter, which encodes the structural proteins. And here I'm showing you this DNA wrapped around a cake, which would be, it is chromatinized. It's wrapped around histone proteins. This would be a nucleosome. Because most DNA, when it gets in the nucleus, is immediately chromatinized by the host cell. So herpes DNA in the particle is not chromatinized. It's not wrapped around nucleosomes as it is for SV40. But as soon as the DNA gets in the nucleus, it's chromatinized. What's the function of that, you may ask? Well, that's a way of the cell from shutting down transcription of foreign DNA. And so viral genomes have to encode antagonists of that. And they most certainly do. So let us go to a quiz at this time. Number 2.
All right, so here we go. Put it on the screen. All right, get y'all thinking caps on. So here you go. Adenovirus is E1A protein stimulating the expression of adenovirus E2 protein, which then stimulates the expression of adenoviruses IVA2 and L4 protein, is an example of A negative autoregulatory loop, B, repression of gene expression, C, cascade regulation, D, dimerization. Adenovirus E1A protein stimulating the expression of adenovirus E2 protein, which then stimulates the expression of adenovirus IVA2 and L4 protein, is an example of A negative autoregulatory loop, B, repression of gene expression, C, cascade regulation, D, dimerization. Even if you don't know what the heck he was talking about, he gave an example of exactly what this question is asking. Don't listen to Chavis unless don't listen to him. I'm going to go back to the question again. Go back to the question. Adenovirus E1A protein stimulating the expression of adenovirus E2 protein, which then stimulates the expression of adenovirus IVA2 and L4 protein, is an example of a negative autoregulatory loop, repression of gene expression, cascade regulation, or dimerization. He said, I feel lonely here. Nobody else want to jump out the window. I'm telling y'all now, you don't listen to Chavis. He told y'all, even if you didn't know, if you're just following along, he gave you the answer.
That's a good one. I'll put that as the last here so I can remember. And we will go back to quiz. What's the result now? 20 of you.
The answer is C.
10 minutes or so, people still.
My bad. The answer is C. Remember, he broke down in the whole conversation, he dealt with Cascade. He didn't talk about those other things like the way he talked about the Cascade. Right here, I scrolled past it. Regulation of transcription by viral proteins, the Cascade Regulation, and then he gave an example of what he was dealing with, right? So, and I'm gonna tell y'all something. Like when I slow this down for myself, I guess, when I slow it down like this and I start this live, you can't catch it. But when you slow it down, man, you pick up on it a little bit more. When he started explaining what's going on here, after you realize that this arrow means promoter, this is an indicator, and this is the DNA, this sequencing and all of this stuff. These are things that are taking place. All right, we get all of that. Okay, we ain't got to get all of that, but we get some of this basic stuff. We know this is DNA. This is the plus DNA. This is the minus DNA. This is a promoter. This is an indicator. Something's transpiring here. Once we've understood that something is transpiring in the sequencing level and amidst the proteins, then there's things that's going there. Look, positive autoregulatory loop may also be negative. This is a regulatory loop that's transpiring here once things go through transcription and it gets to this phase. Boom. So when he comes back and he asked us, just to give y'all the question again, adenovirus E1 protein stimulating the expression of adenovirus E2 protein, which then stimulates the expression of adenovirus IVA2 and L4 protein, is an example of, he ain't talking about repression. He ain't talking about autoregulatory loop, even though this was mentioned, right? But he wasn't talking about that. Dimerization, cascade regulation, because that's what he emphasized in the screenshot. He emphasized this specifically on this slide. Well, let me get to it. I skipped it. On this slide, he emphasized it. He spent too much time right here. And then when he was explaining such, you know what I'm saying? Then it went on. Right, the key word is cascade. But let's go. We're almost done.
Loop, repression of gene expression, cascade regulation, or dimerization. And while you are chewing that over, let's take a look at some questions. I love your questions because to me, the questions are just a great challenge because they're all over the place. They're unexpected. And I like those challenges. Now, the real challenge is finding out where I ended up. If I can remember, and we will go back to quiz. What's the result now? 20 of you dropped out. The answer is cascade regulation. You know, E1A stimulating E2, which then turns on late. That's cascade regulation. It's not a negative regulatory. It's not repression. It's not dimerization. It's just cascade regulation. Straightforward there. All right. I promised I'd show you the cap on mRNA. Remember, the mRNA is synthesized in the nucleus, and very early on, the cap is added, and the cap is present all the way to the end, and it's needed for efficient translation. This is what a cap looks like. So here is base 1 of the RNA. Right there is your sugar linked by a phosphodiester bond to the second sugar, base 2. And base 1 is the base, you know, A or G or C or U or T.
I want to bring this to everybody's attention that he just mentioned sugar twice. So when you're looking at mRNA vaccines, and we just basically break it down and say sugars and salts, what they're doing is compiling the molecular structure of, I'm saying this all wrong. Let me say this the most layman way. Basically, what they're using is their basic understanding of how mRNA is, where the cap is, what's necessarily needed to create, and then the defense mechanism, which would be in this instance of Sars Cov2, the spike. They make the vaccines according to their familiarity with viruses, and then the biological makeup or the molecular structure of what's in us and what's going on through these particular, how I want to say that, these particular, let's just say scenarios of viral behavior.
And these are all 5' to 3' linkages, right? The 5' carbon, the 3' hydroxyl is linked to the 5' carbon. But the cap is backwards. The cap, here's the sugar, here's a guanine base. It is linked in a 5' to 5' way. So the 5' here is linked to the 5' on the capping base. So it's not 5' to 3', it's 5' to 5', plus the 3 phosphates are still there. So that's why a cap is different. 5', 5' linkage, 3 phosphates, and also it's methylated. So this nitrogen is methylated, and these base 1 and 2, the oxygen is methylated as well. So that's what a cap is. That's why it's unique. It kind of protects the 5' end of the RNA, and it allows efficient translation. And the capping occurs co-transcriptionally. Remember, the polymerase binds at the promoter with all its accessory proteins, and it does a lot of abortive transcripts. And only after you get to 20 or 30 nucleotides is the cap added. And I think that's to avoid capping all those abortive transcripts. It would be a waste. And so there's a capping enzyme that associates with the polymerase. Once the polymerase goes a certain distance, it is phosphorylated. 2 phosphates are added to the polymerase that recruits the capping enzyme, which then puts the cap on. So cap is added co-transcriptionally, which means during the process of transcription. I need to talk about the other processing aspects, which are cleavage and polyadenylation. And so let's talk about polyadenylation first. So you make a precursor to mRNA, which is longer than the final mRNA. And one of the reasons is that the 3' end is different, and that these pre-mRNAs are typically cleaved by enzymes and then polyadenylated. So the polymerase goes much farther than it needs to. And this is shown in detail on the right here. Here's a pre-mRNA. There is a site within this mRNA called the polyaddition site. It's typically AAUAAA. And that is recognized by CPSF, cleavage and polyadenylation specificity factor, I believe it stands for. This is a cell protein that recognizes the poly A signal, will then cleave the RNA, recruits a couple of other proteins to do that. So now you've cut, and the right-hand part here is thrown out. Don't need it. And then another enzyme comes in called poly A polymerase, which adds a bunch of A's to the 3' end. So the polymerase makes a long precursor. It's cut at the poly A signal, and then poly A is added. You add about 200 A's to typical mRNAs. So now we can talk about all the different ways to add poly A to viral RNAs. We have post-transcriptional ways that we've talked about today. Cleavage of a precursor to mRNA followed by polyadenylation. It's carried out by cellular enzymes, and all these DNA viruses plus retroviruses do that. And then we have polyadenylation during mRNA synthesis, which we talked about last time. Remember, reiterative copying at stretches of U in the template, that for flu and VSV. Remember, the enzyme stutters at a U sequence, and that's how it makes poly A. So it's during mRNA synthesis. It's not after, as we just talked about. And then for some viruses, poliovirus, alpha viruses, they have long stretches of U in the minus strand, and long stretches of A or poly A in the plus strand. So the viral RNA has a poly A at the 3' end. It's copied to make a negative strand, and all of that A is copied too to make U's, and then the U's are copied back. So it's simply encoded in the genome. Now, splicing is another form of post-transcriptional processing in which these introns are removed. This was first discovered in adenovirus infected cells by two different groups, Richard Roberts at Cold Spring Harbor and Philip Sharp at MIT. They got the Nobel Prize for that in 1993. And the observation was that the adenovirus mRNAs seem to be synthesized first as long precursors in the nucleus. And then when they got out, they got shorter, and people didn't understand what was going on. And so they did an experiment, which is illustrated here. Let me explain it to you. So they took adenovirus infected cells.
Yeah, so Chavis, you're making a good point based on what he said. Absolutely. So this whole lab leak, this is what I mean by let's bring it back. So this is what I mean by the ignorance that people propagate regarding viruses and viral behavior. This is one of the reasons why I'm even interested in this, is because the more that I study the behavior of viruses and how they act, the more that I could analyze the data for myself and come to an honest conclusion based on the data itself and say, hey, this whole lab leak theory thing is even more asinine. When you look at the information or the data that we already have regarding the sequencing and the 96, 97% match of SARS CoV, right, in bats outside of Wuhan and in these other intricate places that are way away from Wuhan, that's an indicator. And then when we have the sequencing going toward the lab, not coming from the lab, that's another indicator. And now you understand the behavior of the virus, the viral behavior. And then he goes down and he tells you exactly what he just said, that viruses naturally do certain things themselves. They transcribe and, and they do. And I'm going to run it back. Let me run it back to that point. All right, listen to him closely.
The mRNA followed by polyadenylation. It's carried out by cellular enzymes, and all these DNA viruses plus retroviruses do that. And then we have polyadenylation during mRNA synthesis, which we talked about last time. Remember, reiterative copying at stretches of U in the template, that for flu and VSV, remember the enzyme stutters at a U sequence, and that's how it makes poly-A. So it's during mRNA synthesis. It's not after, as we just talked about. And then for some viruses, poliovirus, alpha viruses, they have long stretches of U in the minus strand and long stretches of A or poly-A in the plus strand. So the viral RNA has a poly-A at the 3' end. It's copied to make a negative strand, and all of that A is copied too to make U's, and then the U's are copied back. So it's simply encoded in the genome. Now, splicing is another form of post-transcriptional processing in which these introns...
I don't go all the way back to the cleavage site, man.
Talk about all the different ways to add poly-A to viral RNA.
Nah, I don't want to do that. You're going to make me... You're going to make me extend the video out.
That is recognized by CPSF.
We're going to make them... We're going to make them go back to the very beginning of this part, where he explains all of this in this part. I'm trying to get through the video, bro. But when he starts explaining this figure right here, he's telling you right now, he's making a clear indication that scientifically that SARS CoV2 was not modified in any lab.
Black sequences are actually non-coding regions, but they're still joined together to the body. Very important discovery, since this has been found, of course, in many, many viruses, and our cellular genes are largely spliced as well. Splicing can happen in different ways, and its regulation gives rise to different patterns of gene expression. So for example, here is a mRNA with 2 introns, and they can both be removed by splicing to give you the final mRNA with 3 exons, 1, 2, 3. That's called constitutive. You remove all the introns. Sometimes you can get alternative splicing. So this 5' splice site can actually join down here and skip this one. So you could get exon 1, 2, 3, which would be constitutive, but then you could also get 1 and 3. You could skip 2. That's called alternative splicing. You can even get more granular. You could have alternative 5' splice sites and alternative 3' splice sites. So you could get 1 and 2, and you could get different forms because these alternative sites are spaced differently. Here showing at the 5' and the 3' end. So this could be 10 or 20 bases apart, and they could each splice to a different part of 2, and you get slightly different proteins. Now, what happens in adenovirus takes advantage of this, and it's stunning. And here I'm showing you at the top the genome, and here's the major late mRNA, right? There are a number of late promoters, but there's one major late because it makes this huge primary mRNA. And that is processed to give rise to a whole lot of smaller mRNAs that encode different proteins. And the first thing that happens to this long mRNA is that there is poly-A addition at one of 5 possible polyadenylation signals. There's one here at L1, L2, L3, L4, and L5. So each of these is now polyadenylated at a different site, and it's regulated by ways that are somewhat understood. And then these mRNAs can be alternatively spliced in a variety of ways, and some of those are shown here. And you can get different final mRNAs that encode different proteins. And these are just some of the proteins here that can be made. And viral proteins can regulate this alternative splicing. So it gives you a lot of flexibility with a genome, which you may say, oh, we can only encode so many proteins, but actually you could encode a lot more than you think you have. All right, let's do our final question here. Which statement about polyadenylation of DNA virus mRNAs is correct?
All right, so which statement about polyadenylation of DNA viruses mRNAs is correct? And you just explained it. So it always occurs in the cytoplasm. It occurs after the cleavage of pre-mRNA. Polyo-A is added at the 5' end of the pre-mRNA. It's specified by a stretch of U residues in the templates. This one's a little tricky for a bunch of people because, you know, he really went off the cusp. He didn't go by the book on this one. So I'm going to kind of slide back through here real quick to try to help everybody out. Again, the question is, which statement about polyadenylation of DNA virus mRNAs is correct? It always occurs in the cytoplasm. It occurs after cleavage of pre-mRNA. Polyo-A is added in the 5th end of the pre-mRNA. Is specified by a stretch of U residues in the template. All right, y'all ready? Make sure that you're paying attention. When he was explaining this, where was everything happening at? Let me zoom in. What was the cap transpiring? I ain't gonna take too much longer. I'm trying to give you an indicator of really what he's saying in layman. The modification of mRNA cleavage and polyadenylation was his question. Y'all gonna get stuck at the arrows, I know it. So I think y'all might have seen the answer now. Skip the splicing. Skip the proteins and mRNAs. Question is bigger. Which statement about polyadenylation of DNA viruses, mRNAs, is correct? It always occurs in the cytoplasm, A. It occurs after the cleavage of the pre-mRNA, B. C, polyo A is added at the end of 5' of the pre-mRNA. Or D is specified by a stretch of U residues in the template. That boy DeMar guessing. Everything going with he guessing, he going with B. Man, I can't make it no more simpler. Ah, shoot. The pseudo jumps in and says F. Again. Which statement about polyadenylation of DNA virus mRNAs is correct? It always occurs in the cytoplasm. It occurs after the cleavage site of pre-mRNA. C, poly A is added at the 5th end of the pre-mRNA. Is specified by a stretch of residue in the template. I just helped you out to where you should have at least have narrowed down to Zay, boy, Zay crazy. I just helped you narrow down 2 things. So I'm going to go back to it because I just helped you get rid of 2 answers. Because it says which statement about polyadenylation of DNA virus mRNAs is correct. It always occurs in the cytoplasm. It occurs after cleavage of pre-mRNA. Polyo is added at the 5th end of pre-mRNA. Is specified by a stretch of U residues in the template. Let me see who else answered. 2 people said B. 1 person said DC. All right, so let's see what the answer is, man.
A, it always occurs in the cytoplasm. B, it occurs after cleavage of pre-mRNA. C, poly A is added at the 5' end of pre-mRNA. And D, it's specified by a stretch of U residues in the template. Let's go to our questions here.
Told y'all I got rid of 2 of them when I went back to the
I
chart.
didn't show the quiz when I explained the answers. Let me go back and do that. I'm sorry. I have to push the button. Which statement about polyadenylation is correct? The answer is it occurs after cleavage of pre-mRNA.
All right. So when I went back to let me show you what I did. When I went back to here, right? When I went back here, I eliminated the stretches of U and template RNA. I got rid of that. Look, got rid of this stuff. If we go look at the answer, you only had two choices. You already know when I went back to the other grid to show, I got rid of this one, D for you. Then I went back to the example that he used where you could see the fifth end. It was later on the diagram. So let me show you that. When he was here, look what the fifth end is. But what he was talking about when talking in regard to the question asked, it's later on. So we're not talking about that. So it occurs after the cleavage of pre-mRNA. So where's the pre-mRNA located? Does anyone know where the pre-mRNA is located? We got to go back. All right. I went too far back. Oh.
And very early on, the cap is added, and the cap is present all the way to the end.
Look at Zion over there.
For efficient translation. This is what a cap looks like. So here is base 1 of the RNA. Right there is your sugar linked by a phosphodiester bond to the second sugar, base 2. The base 1 is the base, A or G or C or U or T.
Look, so even here it says endonucleatic cleavage poly A addition, splicing, export, mRNA, export, cytoplasm out here. So the cytoplasm is out here. So we go back to the question, right? It occurs after cleavage of pre-mRNA. After the cleavage, it always occurs in the cytoplasm. It couldn't occur in the cytoplasm because everything that we just looked at as far as how it functions in the example here, in this right here, right? The cytoplasm out here. The other stuff is happening in here. See, this is basically the first line of RNA. This is the pre-RNA. This is after the fact. See? You got your mRNA here, and then it comes out. So when he asked the question, the way that he asked the question was based off of something that wasn't in the book, but he was just going directly off that and asking us that. So which statement about polyadenylation of DNA virus mRNAs is correct? It occurs after cleavage of pre-mRNA. It wasn't at the cap site. Polyo A is added at the fifth end of the pre-mRNA. That's the cap site. Nah, I wasn't capping. We weren't capping in this rap. Now he goes on to have a little conversation about some other stuff here. Not really going to get too much into that. I just wanted to get through the 3 questions and pretty much sum up the study group for the day. So again, look at Zane. It was Zane right there. Zane in the building. I'm in the building. Look, Unc in the building. You know what I'm saying? Unc gonna text me and ask me where I'm at. I'm already in here answering questions. But that's pretty much it for that, man. You know what I'm saying? I do want to thank everybody for tuning in to the study group. Like I said, we all learning here, and I appreciate everybody's time. We took a 2-hour video, made it 3 hours, but it is what it is, man. Like I said, we all studying to learn and improve and get better every day. So I don't want to hold nobody up, keep everybody abreast of something else they could be doing. I do appreciate everybody tuning into this episode of Transcription and RNA, our part 3 of our class. And hopefully I'll see some of y'all tomorrow, man. But y'all already know, go pick up your copy of What Sars Cov2 Taught Me. Same Tree, Different Branch Publishing. Go pick up your copy of What Sars Cov2 Taught Me. ASAP, get your copy. I got 5 books on the way to New York from one person. I think somebody else just purchased the book. But I think it's the reason why somebody just text me and want me to holler at them about something. Yeah, somebody, okay, all right, cool. So, look, man, I appreciate everybody for tuning in. Y'all enjoy y'all evening, man. Watch the rest of football. I think it might be halftime or whatever. Enjoy some foolery or whatever. Again, pick up your copy of What Sars Cov2 Taught Me. Order your copy right now, man. Maybe we'll get into chapter 1 of that book. But pretty much we're doing what we need to do to understand this whole pandemic, the origins of the virus and the whole 9. And it's going to take a collective effort. So teams win. That's how we rock and roll. Again, y'all enjoy y'all evening. And y'all already know what I'm about to do.